Hello, good morning, afternoon, and evening to all of our viewers tuning in from different time zones across the world, and a very warm welcome to the platform of Biologically Speaking. We are back with yet another installment of our webinar series, and today, on the occasion of World Population Day, we have a very special guest with us. We have Professor Amitabh Joshi from Jawaharlal Nehru Center for Advanced Scientific Research, Bengaluru, India. Professor Joshi has been one of the true stalwarts of the Indian academia and in the field of evolutionary biology. After completing his doctoral degree from the Washington State University, he worked with Dr. Lawrence Muller at UC Irvine. Over this tenure, he studied topics like coevolution and variation in competition and adaptation to larval and adult crowding using fruit flies as a model system. He joined JNCASR as a faculty in 1996 and heads the evolutionary biology lab there. His research focuses on the interrelations between the genetics of organisms and their ecology and the influence of these factors in their evolutionary path. His laboratory studies various evolutionary aspects such as the rate of development, patterns of progeny and aging, and investigate the development of competitive abilities and stress resistance in organisms. Dr. Joshi's work has synergized evolutionary studies with theoretical work using computer simulations. His exemplary work has not only been published in topmost research journals like Science, but also earned him innumerable accolades. He has been elected fellow to the Indian Academy of Sciences, Bardeen Institute for Advanced Studies, National Academy of Sciences, India, and has also won the Shanti Swarup Bhatnagar Prize for Biological Sciences in 2009. Not only a great researcher, but Professor Joshi has also been a true connoisseur of poetry and philosophy in life. To quote him, while poets and mathematicians have the luxury of creating alternative universes, biologists must make do with model system. On that note, I would like to pass on the mic to Professor Joshi and let him tell us all about his learnings about population ecology from Drosophila model system. Professor Joshi, the screen is all yours.
Thank you very much, Suradeep, for a very kind introduction. And again, good evening, good morning, or good afternoon, as the case may be, to all the viewers. Before I start, let me just express my thanks to the entire organizing team at Biologically Speaking for giving me an opportunity to share some of our work here on this really wonderful platform. So I am by training a geneticist, and most of the work we do is actually in experimental evolution. We also work in experimental population ecology, and I chose to focus on that for today's talk because this is also coinciding with World Population Day. So what I'd like to do, if I can get my screen up, is my screen coming up?
No, it's actually showing blank. Maybe you can share this.
I reshare again.
Yeah.
Just a second. I'm sorry.
No worries. Yeah. No worries.
Wow, it's not giving me that option.
Do you see the share button at the bottom?
Yeah, I got to it, but in the drop-down list of screens to share, it's not showing me that particular screen anymore. It's showing me everything else that happens to be open.
Maybe close the file and then try to again open it.
This is really weird. Um, this is bizarre. I don't know what's happening. It is not giving me the option to share the PDF screen at all. It's showing me even screens which I've shut down are still showing.
One thing you can do, you can send us the PDF and we can try opening it from our side.
Okay, let
Yeah.
me do that. So sorry about this.
No worries.
Okay, I've sent you the file. My apologies. It worked in the trial run. I have no idea. Oh,
Yeah.
now I get a little message saying your browser has blocked your screen. Click the screen icon in your address bar to allow access and reload the page.
Yes, we will try to share it from our end, and in the meantime, if you can work it on your end, that would be excellent. But
I don't know what's going on. This anyway. Let me just start with some general remarks while you get the screen going. So as I was saying, most of our work is actually in evolution, but we also do work on population ecology, and these lines of work intersect in certain ways also. So many of the themes I will talk about today are actually on the interface of ecology and evolution as well. In particular, I will be talking about things like the evolution of population stability, um, whether the evolution of competitive ability and the ability to withstand high levels of crowding actually affects the kind of population stability that those populations will then show and stuff like that. I will also be talking a little bit about stability itself in populations with and without spatial structuring, and look at aspects of stability like constancy and persistence, and we'll talk about what these are as the slides manage to get available. One of the things I'd like to stress, just for those younger people in the audience, is that population ecology, interestingly, is something I never formally studied when I was a student or grad student. I got interested in this when I was a postdoc, and ever since I've been doing research in it and teaching it. And I mentioned this just to say that, if you come across a line of research that you find interesting, one should not feel shy of saying, oh, well, I don't have much of a background in this. One can always acquire the necessary knowledge and tools to deal with it. So one should feel free to go ahead and get into things that seem interesting, even if one has no particular background in that. Thanks a lot. I finally do see the screen. Maybe it would help if you could make this full screen. It'll be in view. You will get the full screen option under view. Full screen. Yeah. Wonderful. So could we have the next slide, please? So before we start with the work, I just want to briefly say something about what population ecology really is. So these are just three representative data sets of how many individuals there were in the population at various points in time. This happens to be a data set from Japan. It really doesn't matter. I just want to make the point that if you look at these data sets, you notice that the numbers have a certain behavior over time. There are some similarities for all these three species, but there are also some differences. And what population ecology is largely about is trying to understand the factors that shape how these numbers of individuals in a population change over time and what kinds of factors drive the similarities and differences among populations. And what you can appreciate is that if one really wants to tease apart the causes behind similarities or differences in patterns of dynamics of this kind, it's very difficult to do with natural populations in the wild because there are many, many environmental factors that are not in one's control. And so it is easier to address some of these more fundamental questions in population ecology using well-replicated and well-controlled systems in the lab. Next slide, please. And this is in consonance with the sentiment expressed by my favorite poet, Allama Iqbal, who said, Guftan jahone mo oyo batumi sozad, Guftam ke nami sozad, Guftan ke barham sad. So God asked me if I did find his universe to my liking and taste. When I said no, then he replied, create your own and lay this waste. And again, as Shoradeep mentioned, so poets and mathematicians really do have the luxury of creating their own beautiful alternate universes. We have to manage with model systems. Next slide, please. And the system we use is Drosophila melanogaster. We do occasionally work with some other species of Drosophila also. And the main advantages of the system is it has a reasonably short generation time. So you can do multi-generation studies. You can maintain them in large numbers without too much problem. They are small. Most importantly, the basic ecology of laboratory cultures of Drosophila is very well understood. And that is very helpful because we are drawing on something like 70 years of understanding of how Drosophila cultures tick. Controlled manipulations and perturbations can be carried out to the populations to look at the responses. And population level replication can be done, which is most important compared to field studies. And finally, this kind of work actually doesn't require any fancy equipment or large budgets. It's fairly cheap. It is labor intensive, but not particularly expensive. Next slide, please. So before I go into the work, I thought I would talk a little bit about the history of where this work is coming from and how different lines of work in the past have sort of intertwined to give this. So time in this slide is running roughly from top to bottom. The topics below the red line are ones that I will be sharing some research results with you about. The topics above are things that led to this work. And the topics highlighted with a red star are things we are presently working on. Those highlighted with a blue star are ones which I have been involved in the past. And one point I would like to make here is that we are all in biology very familiar with Drosophila as a model system for genetics, thanks to the work of T.H. Morgan and others after that. What is often not realized is that even in population ecology, Drosophila was used in the early 20th century by people like Nicholson and others. And at that time, the initial studies were more about using Drosophila cultures to try to fit simple models of population growth that were just being developed and estimate their parameters and so on. There were also many studies of lab ecology that tried to establish what is the basic ecology of a typical Drosophila culture. Eventually, these coalesced into studies of competition and density-dependent selection, which is the idea that what kinds of selection pressures a population faces and what kinds of traits are selected for on a population. varies with the levels of crowding that population chronically experiences. And eventually, in the 1980s and 90s, many of these strands coalesced in the extremely seminal work of Larry Muller at UC Irvine, and before that at Washington State and UC Davis. And this was work on trying to look at how fruit fly populations evolve adaptations to crowding if you force them to undergo crowded conditions every generation. And he also came up with a very detailed model that I'll talk a little bit about. And that model of his in 1988 really is the linchpin which permits the kind of work that we were able to subsequently do and the work that I'm going to be sharing. Next slide, please. So, thanks to a lot of people who studied the basic ecology of Drosophila cultures in population growth and regulation in Drosophila cultures, we understand quite well what are the major density dependent feedback loops, the negative loops that are responsible for population regulation and therefore population dynamics in Drosophila. So the middle column in this schematic shows you the progression during the life cycle from eggs to larvae to pupae to adults, and then again adults lay eggs and the next generation starts over. The photograph on the left shows you a typical Drosophila vial. So these are like small test tubes which can have up to a couple of hundred flies in them. And in most of the talk I'm going to give subsequently, when we're talking about a population, we are talking about a single vial population. Now the dashed arrows on the left of the schematic as they appear on your screen are indicating the three density dependent feedback loops. You have larval mortality caused by larval density. If there's too much larval crowding, some of the larvae don't get enough food to sustain complete development to adulthood by the time food runs out, and so mortality goes up. Also, when there is larval crowding, many larvae do survive, but they get less than an optimal amount of food. So they are able to become adults, but they become small adults. And small flies, small female flies, all else being equal, lay less eggs. So there's also a negative feedback of larval crowding on adult size. And finally, there is a feedback of adult crowding on the fecundity of those adults based on whatever their size would be. So over and above the size effect on fecundity mediated through larval crowding, there's also a direct adult crowding effect on fecundity. And a lot of work over almost 60 or 70 years led to the understanding that principally the dynamics of population numbers in a Drosophila culture is dominated by these three feedback loops. Next slide, please. So this is the paper I mentioned, 1988. The model is given here for our present purposes. The details of this model are not important. I just want to draw your attention to 2 or 3 points about this work. One is that this was the first model of Drosophila population dynamics that took into account explicitly all of these three feedback loops that I just mentioned. So all those are built into this model. And the second thing is that this model gave rise to certain very interesting predictions, some of which opened up the path for future experimental work. So one of the theoretically important predictions from this model was that, contrary to what was believed at the time based on much simpler models of population growth like the logistic or the Ricker model, this model indicated that it was possible to actually evolve to become more competitive without necessarily altering the so-called carrying capacity or the equilibrium population size of the population, or indeed without necessarily evolving a smaller body size. At that time, that was the dominant belief in the community, that adaptation to crowding meant you basically evolved to become smaller and more efficient, and that gave you, among other things, a higher equilibrium population size. And this model really showed that there are many other ways to become more competitive under crowding conditions, at least in principle. And subsequent work showed that actually there are many ways in which you can become more competitive, but that's not the theme of today's talk. The practically really important prediction coming from this model was that if you look at two kinds of food regimes, You can have an LH food regime, which means low levels of food provided to the larvae. That's photograph uh, A and C on the top, where you have low levels of food for larvae but high levels of food for adults below that. This kind of regime was predicted to induce large fluctuations in population size every alternating generation or a 2-point cycle. By contrast, an HL food regime, which combined high levels of food for larvae and relatively low or not so rich levels of food for adults, was predicted to give rise to relatively stable dynamics with some noise around a point or a putative point equilibrium. Next slide, please. So these are data from the first attempt to test these predictions about the effects of these two kinds of food regimes using very large populations. These are cage populations of Drosophila. As you can see on the y-axis, population numbers are in several thousands. It's not terribly important for this talk what the identity of these populations is. So there are 10 populations under the HL or stabilizing regime, 10 populations under the LH or destabilizing regime. And you can see that indeed the populations in the destabilizing LH regime do seem to show, even visually, very much more unstable dynamics than the populations on the left columns. So at this point, this was work I was involved in towards the end of my postdoc in 1995. And one of the things we were interested in doing when I was starting my lab in Bangalore was to look at metapopulation dynamics and try to test hypotheses about stability and dynamics in metapopulations. So metapopulations, we will come to this in more detail later on. They are basically sets of populations connected by some amount of migration, or as it is nowadays called, dispersal between them. And so in order to set up metapopulations, we wanted to work with local individual populations that were quite small. And so we wanted to check whether this prediction about HL and LH food regimes giving us the ability to experimentally produce unstable or stable dynamics at will would actually hold up in really small single vial populations in the face of greater demographic stochasticity. Next slide, please. So these are data from one of the first experiments we did around 1997 in our lab in Bangalore. So we took single vial populations of Drosophila, subjected them to either the HL or LH food regimes. The time series of the population sizes are shown on the two graphs on the top, on the left and right. And below that, the two smaller graphs are showing you autocorrelation spectrums and Fourier analyses on those time series. And again, without going into the technical details of what these are, if you look at the autocorrelations under the LH food regime, they show a zigzag pattern of alternating negative and positive autocorrelations across time. That is a signature of 2-point cycles. Similarly, under the LH regime, the Fourier analysis shows a contribution peak of a frequency of 0.5, which corresponds to a period 2 cycling behavior, whereas there is no such evidence of cycling in the autocorrelation spectrums or the frequency contributions of different frequencies for the HL food regime. So indeed, it was heartening to see that this tool does give us the signature unstable or stable dynamics, even in very small populations, single vial populations, where demographic stochasticity is going to be high. The table at the bottom may or may not be very clearly visible. I'm sorry if it's not. I'll just summarize what the take-home message from that is. So what we did was we took these time series of data and we fitted a simple 2-parameter model of population growth, the Ricker model, which has 2 parameters, R and K. R is the maximal possible intrinsic per capita growth rate at very, very, very low densities. And K is the equilibrium population size. And what we were able to see is that regardless of whether we did the fitting based on census data on just the total number of individuals, or whether we did the fitting accounting for how many of the individuals were male and female, because only females produce new individuals for the next generation, it didn't affect the degree of fit substantially at all. So this was also helpful because it suggested that for bigger experiments, we could just go with total census numbers, in fact, instead of having to do a sex-specific census, and that saves a lot of time and becomes really important for metapopulation experiments for a variety of technical reasons. So next slide, please. So now let me introduce some of the kinds of questions that I will share results with you about how we try to address these. So one is, are these two aspects of stability in population ecology, that is constancy and persistence? I'll come to what these are in the next slide. Do constancy and persistence correlate with each other? Do they co-evolve together? So at the time when we started this work, the general belief in the community was that constancy and persistent stability are sort of two sides of the same coin. So if a population has unstable dynamics in terms of constancy, it will also be unstable in terms of persistence. In other words, it will be more extinction prone. We also wanted to test predictions about whether simple perturbations in terms of immigration, emigration, adding or subtracting individuals to the population can stabilize populations with regard to constancy and persistence. And again, whether constancy and persistence respond similarly to these perturbations. The third question we will be looking at is, does population stability evolve? Either through life history evolution or through density dependent selection. And again, I'll talk about the backdrop of this when we come to that specific study. And finally, a couple of questions about metapopulations. First, how do local dynamics and migration or dispersal rates interact to affect synchrony among the subpopulations or the local populations? And how do local dynamics and migration or dispersal interact to affect local and global constancy and persistence in metapopulations? And again, these are very fundamental questions in population ecology. There's a lot of theory addressing these things, but these are very, very difficult, if not impossible, to actually study with wild populations, partly because of the difficulty in doing manipulative experiments and perturbations, partly because of the difficulty of just doing a good census, and partly because of the problem of not being able to replicate entire populations and metapopulations, holding many aspects of the environment and the genetic composition of the populations constant. Next slide, please. So constancy stability, this is a differentiation that was introduced by Grimm and Wissel in a very major paper in Ecology about 30-40 years ago, to bring about some consistency in how we use the terms. And they basically pointed out that when ecologists or population ecologists talk about stability, they usually either mean constancy, which is the tendency of populations to show relatively small fluctuations in size from generation to generation or time step to time step. And persistence refers to the tendency of populations to not go extinct over a given time span. Persistence is usually expressed as the inverse of the probability of extinction in a given time span. Constancy, at least more recently, is now routinely expressed as the fluctuation index, which was a measure that we had proposed as a replacement to looking at the coefficient of variation, because this has some slightly better properties. So the fluctuation index is basically the average absolute magnitude of one step change in population size over time, scaled by the average population size over the time series. Next slide, please. So the first study I want to talk about has to do with testing some hypotheses about constant number immigration. That is the input of a constant number of individuals from outside into a closed population. And theoretical work from many people around the world had suggested that constant immigration would enhance constancy stability in populations that had unstable dynamics where numbers were fluctuating a lot. So we set up Drosophila experiments with 3 treatments: a control which just had a bunch of populations with no immigration, a low migration treatment in which we additionally introduced 2 females into the population from outside, and a high migration treatment in which we introduced 5 females per generation per population from the outside. The generation was run for 17 generations and census data were collected every generation. So in all the work I'm going to show you, the primary readout is basically the census data per population over time. And there were a total of 48 single vial populations. 24 of them, 8 replicate populations in each of the 3 treatments were in an LH regime, and 8 were in, again, a stabilizing or LL food regime, which combined low levels of food for larvae with low levels of food for adults. So LL, like HL, is also a stabilizing regime. This little graphic on the left just shows you the raw time series data for what it's worth. It's not important. Let me focus on the results in the 3 graphs on the right. So the top graph on the right shows you the mean number of extinctions over the course of 17 generations in the LL populations in the black and in the LH populations in the white circles. And along the x-axis are the controls, the low migration and high migration. And what you can see from the top graph is that indeed the number of extinctions came down compared to controls in the LL regime, but not in the LH regime. In the destabilizing regime, in fact, there is a difference between low and high migration in terms of the number of extinctions. So persistence stability is actually becoming negatively affected by going from low to high migration, whereas constancy persistence, the second graph on the right-hand side, is enhanced because the fluctuation index comes down when you go from low to high migration. So this was one of the early indications that constancy and persistence are actually responding independently to a certain perturbation predicted to enhance stability. So these 2 aspects of stability don't necessarily respond similarly to the same perturbation. Next slide, please. Okay, let me now shift to the issue of the evolution of population stability. This has been an issue very dear to our heart. A lot of the work on this issue has actually been done entirely in our lab over the last 20 years or so. The reason this was a conundrum in population ecology was because of the remarkable work of Robert May, theoretical work in the 70s and onwards, which suggested that in simple models of population growth, if you enhance the maximal per capita growth rate, or R, you get fairly unstable dynamics, indeed going up to chaotic dynamics if you increase R sufficiently. Now, the point is that the kinds of biological traits that feed into higher R are things like higher female fecundity, better survivorship up to adulthood, and things like that. These are all traits that, all else being equal, you would expect natural selection to increase over time in most populations. So, given the work of Robert May, one could naively expect that most populations will actually show fairly unstable dynamics, especially if there are not other factors like predators that are tending to stabilize them. But if you actually look at single population dynamics in the lab and in the wild, stable dynamics are extremely common. So the question then began, and theoreticians looked at this, as to what might be the mechanisms by which population stability evolves. And one class of theories basically invoke the idea of life history trade-offs. So life history refers to things like the duration of the juvenile phase, the timing of reproduction, the magnitude of reproductive output, the adult lifespan, and things like that. So these are all traits that feed into both Darwinian fitness on the one hand and into maximal rates of population growth on the other. So there were many models that suggested that stability might evolve because many of these life history traits trade off with one another. And so if you have selection that favors one life history trait, it could cause a correlated decrease in something else, which might as a byproduct give rise to population stability. So we tested this with 2 sets of populations we had in our lab already. We had 4 populations selected for over 100 generations at that time for rapid egg to adult development, as a result of which they also became much smaller as adults, as you can see in the little picture on the top. So these are male Drosophila on the left from the selected populations, and on the right are male Drosophila from the ancestral controls. And we predicted that because the FEJ flies, the selected flies, have become so small, their fecundity perhaps has gone down. We had evidence that their pre-adult survival had also gone down, so perhaps they would have evolved to become more stable. So we derived small single vial populations, did a population dynamics experiment with representatives from all these populations in LH and HL food regimes. And the expectation was that FEJs might be showing greater constancy, and we also wanted to look at whether they showed greater persistence. Next slide, please. So the data on the left, the top graph is showing you the fluctuation index in the LH and HL food regimes. The selected lines are the white bars, and the fluctuation indexes are indeed substantially lower than those in their ancestors. So they have become more stable with regard to constancy. This is mirrored in a decrease in the graph just below that on the left, which is the intrinsic growth rate, R, the maximal growth rate. And again, the R values estimated from the time series for the selected populations are substantially lower than their ancestral controls. On the right-hand side are some probability of extinction plots. I don't want to go into the details of this particular story. I just want to point out that in this particular experiment, what we found was that the selected populations, the FEJ populations, did not show decreased extinction or a enhanced persistence, even though they showed enhanced constancy. So this was actually the first study showing that constancy and persistence do not necessarily co-evolve. And this was really the first time anybody tried to test any of these models of the evolution of stability. And I'm very happy to share with you that this particular work, which was done by two students and a postdoc, Mallikarjun Shakarat, N.G. Prasad, and Sutirth Day, at the time, was recently included among 65 studies starting from Darwin to the present time in a book that came out last year called Conceptual Breakthroughs in Evolutionary Ecology. And this is pretty much the only study from outside the Western world that found a place in that collection. So we're very thrilled about this. Next slide, please. The second aspect of the evolution of population stability that we were interested in, also given our interest in the evolution of adaptations to crowding, was that again a lot of theory coming from density-dependent selection suggested that stability might evolve as a result of adaptation to crowding due to an RK trade-off. So there was this notion, which has some empirical support in many populations of Drosophila from earlier work by Larry Muller, that maximal intrinsic growth rate, or R, of these simple population models and carrying capacity or equilibrium population size K trade off. And what happens therefore is that as a result of adaptation to crowding, populations might be expected to evolve traits that contribute to a greater value of K, which automatically causes a reduction in the value of R, and therefore results in increased constancy. Now, one earlier study, the CAGE study, for which I've shown you data with really large populations, found no greater constancy in crowding adapted populations at all. We decided to look at this again in some populations of crowding adapted Drosophila ananaceae and Drosophila melanogaster, because these particular populations in our lab had, like the earlier populations that Larry Muller developed, they had become more competitive in response to being forced to undergo crowding each generation, but they had evolved adaptations to crowding and competitive ability that were very different in terms of the traits involved than the earlier populations in Larry Muller's lab. And we looked at these and indeed found, if you look at the top graph on the right, so the white bars here are the ancestral controls, the AB populations, and the ACU are the crowding adapted ananaceae populations. The mean fluctuation indexes were reduced and therefore constancy stability enhanced in the selected populations that were adapted to crowding. And in this particular case, the mean number of extinction events also went down, and they did indeed, in the third graph you can see, they did indeed maintain a higher mean population size. So we also got very similar results from our melanogaster populations, which I'm not showing here. So it turns out, again, to summarize what is a really long and nuanced and actually very interesting story, it turns out that if you look at populations that have evolved to become more competitive as a result of chronically being subjected to larval crowding, it turns out that how they evolve to become more competitive, which traits they evolve, actually depends on fairly nuanced ecological details of how they experience the crowding and which traits they evolve the enhanced competitive ability through. Actually, those determine whether or not there will be consequences for stability. So the picture is not as simple as simply saying that you adapt to crowding and you will become more stable. Next slide, please. Okay, so let me now shift gears and for the remainder of the talk a little bit about some of the studies on metapopulations. So as I said earlier, metapopulations are a set of populations that are independent enough to have their own independent population dynamics, which we will be referring to as the local dynamics of a local population or a subpopulation. But there is also some amount of movement or migration or dispersal between them, which potentially can synchronize to some degree their dynamics. So this has a lot of theory behind it, much of which actually is published in the physics literature under the general heading of coupled oscillators or coupled map lattices. And the idea here again is why we got interested in this was that different models of these kinds of coupled populations or metapopulations suggest a variety of predictions for the impact of altering migration rates. One general observation, point number one below, is that if local dynamics are unstable or indeed chaotic, subpopulations will eventually tend to be fluctuating out of phase. And this will, of course, be stabilizing in terms of constancy for the entire ensemble due to an averaging effect. But then comes the point of now, if you increase the migration, what will happen? Some models indicate that migration would synchronize subpopulations, thereby destabilizing the entire ensemble, because now all of them will start fluctuating in phase. And if all of them are fluctuating in phase and the troughs are quite low, the entire set of populations could have a high risk of going extinct. Other models suggest that migration in some cases, especially if density dependent, might actually act as a damping factor to damp out the individual fluctuations in the dynamics of local populations. And this would stabilize both the local populations and the entire metapopulation. There are also models which suggest that migration rate changes will not affect subpopulation dynamics in any major way. Now, the problem is that these are all decent models. There's nothing obviously very unreal or very wrong about any of them. And so we decided that now that we have the experimental tools in hand to try to set up studies to test some of these issues more rigorously with our fly system. Next slide, please. So these are data from a simulation study. So what we are doing is we are taking the simple Ricker model of population growth, which has these two parameters, R and K, the maximal per capita growth rate, which is also the bifurcation parameter that determines the dynamics of a single population following this model. And we are modeling little metapopulations with 9 subpopulations each that are connected by nearest neighbor migration. And we are doing the simulations for two kinds of metapopulations with high migration, 30% migration each generation to the two neighboring adjacent local populations. Those are the red dots in the graph, and the green dots are the low migration treatment in the simulation, which is 10% nearest neighbor migration every generation. And what is plotted on the y-axis is the cross correlation coefficient. That is the coefficient on average across space between the population size of neighboring subpopulations, adjacent subpopulations. So this is a measure of synchrony between the dynamics of the local populations. Positive values of cross correlation indicate positive synchrony, they are fluctuating in phase. Negative values of cross correlation coefficient indicate asynchrony, or that they are fluctuating out of phase with each other. And we are looking at cross correlation coefficient as a function of increasing the maximal per capita growth rate R in the simulations. Now, what we see from the results of these simulations is that at low values of R, before we enter into the extremely complicated dynamics of chaotic regime, which is around 3, prior to that, cross-correlation coefficients are reasonably high, and more importantly, do not differ between the high and low migration treatments. You have positive synchrony. Once you get into high or potentially chaotic values of R, we expect from the simulations the high magnitude migration treatment to still give you positive synchrony, whereas the low migration treatment begins to give you negative or asynchrony or out of phase fluctuations. And now again, our destabilizing or LH treatment and our stabilizing or LL treatments allow us to test this prediction, because the R value of LH treatments is around the range of little over 3, where the prediction is very different for cross correlation between high and low migration. And the R values estimated from data on LL populations are around 2.3 to 2.4, where the prediction is no difference between low and high migration and generally high values of positive synchrony. Next slide, please. So this is the experimental setup for the first experiment I'm going to describe. We have on the left these 9 single vial populations. The double-headed arrows are to indicate that each population in principle exchanges migrants at a constant fraction of its population size every generation with its 2 immediate adjacent neighbors. So this is nearest neighbor migration at a rate of either 10% per generation or 30% per generation. The control just has 9 independent populations with no migration between them. So we have 9 patch metapopulations, 3 treatments, 4 replicate metapopulations in each treatment, each migration treatment, and the entire experiment is replicated with food regimes corresponding to 2 types of local dynamics, LH or destabilizing the local dynamics, and LL or relatively stabilizing for the local dynamics. Next slide, please. So these are the data first from the LH regime experiment. On the left panel, on the top, you just have population numbers on the y-axis versus generations on the x-axis, and this is for total metapopulation numbers. And you can see that both the controls and the 30% migration metapopulations, overall, the entire metapopulation undergoes much more fluctuations over time than metapopulations experiencing 10% migration in the middle. You can eyeball this. On the top graph on the right are plotted the mean fluctuation indices of metapopulations, and this captures the result shown in the time series on the left. The lowest fluctuation index and therefore the highest constancy stability is shown by metapopulations subjected to 10% migration. However, this is not because 10% migration is damping out the local dynamics, because if you look at the second graph on the right hand side, this is the mean fluctuation index for subpopulations within each kind of metapopulation. And in fact, the subpopulations, the local populations in 10% migration have higher fluctuation indices and therefore less constancy stability than either the controls or the 30% migration metapopulations. What is going on is clearly shown in the bottommost graph on the right. This is plotting the cross correlation coefficient for these 3 kinds of metapopulations. And the control and the 30% migration metapopulations show positive synchrony, whereas the 10% migration metapopulations show negative synchrony, or they are fluctuating out of phase, and that is why the time series is getting damped out by an averaging effect. So this particular experiment was published in 2006, and this was actually the first experimental verification of the theoretical prediction that moderate levels of migration are likely to stabilize dynamics by making the local populations go out of phase with each other. Next slide, please. These are the results from the corresponding experiment done, but with all the subpopulations subjected to the LL or stabilizing regime. And what you see here really is that nothing much differs. If you look at the top left graph, it plots the mean metapopulation fluctuation index. There is not much difference in constancy stability across the three treatments. The graph on the bottom left plots the mean subpopulation fluctuation index. Again, there is no difference in constancy stability at the local population level across the three migration treatments. The top graph on the right is showing you synchrony, or the cross correlation coefficients in the three treatments. And the main point to note here is, although there is a difference in magnitude between the positive synchrony experienced under high migration versus low migration, but low migrations are still showing essentially positive or maybe close to zero rather than negative synchrony. There is no negative synchrony at 10% migration unless the intrinsic dynamics are sufficiently unstable. And the bottom graph on the right is basically showing you the mean number of local extinctions. And this just illustrates a general phenomenon called metapopulation rescue, which is that when you have migration as opposed to no migration as in the controls, it tends to reduce the propensity of local populations to go extinct. So the important take-home message from this particular experiment was really that migration rate interacts with the nature or stability of the local dynamics in affecting synchrony and local and global dynamics and stability, either in terms of constancy or in terms of persistence. So one can't really make generalized statements about the impact of migration. One needs to qualify it with some understanding of what the local dynamics in the individual subpopulations are, because there are these strong interactions. And again, teasing apart these interactions would really not have been possible if one was trying to do these studies with wild populations. Now, one last experiment I'm going to come to is based upon the idea which I alluded to earlier when I was showing you what a metapopulation is. And this is the theoretical prediction that if you have a combination of really unstable local dynamics in terms of constancy, so you have large fluctuations at the local population level, and you, for some reason, some ecological reason, you have high coherence or synchrony, then the entire metapopulation will start oscillating violently and will come down to very low numbers every other generation, and that could increase the risk of extinction of the entire metapopulation. And so the last experiment that I'm going to look at is an attempt to test this prediction. Next slide, please. So here we have similar metapopulations, except that for simplicity, we switch to a 6-vial metapopulation. So there are 6-vial metapopulations with nearest neighbor migration between adjacent subpopulations. There are two levels of migration treatment applied here, 10% low migration or 30% high migration. And then in addition, within each migration treatment, we have 3 kinds of perturbation treatments. There is a control in which we are not perturbing the metapopulation at all. We are just applying whatever migration treatment those metapopulations are supposed to get. Then we have 2 treatments where we are perturbing. And the perturbation here is what is often called pinning in the physics literature. There's a lot of theory about the stabilizing effect of pinning on local and global stability in coupled map lattices. Uh, so we are choosing to pin by adding 10 extra flies each generation. That is the nature of the pinning here. You are from outside the system adding 10 flies. In the block perturbation, in the middle, what we are doing is that we are doing the pinning onto two adjacent subpopulations. So once these populations have received these 10 extra flies and then they are subject to the natural migration that happens in the metapopulation, some of these pinned flies can spill over to two more adjacent populations. So the pinning in principle affects totally 4 of the 6 subpopulations in the block perturbation treatment. Whereas in the scattered perturbation treatment, we pin opposite populations in this design. And so when the pinning can trickle down to adjacent populations as a result of the regular migration, all 6 subpopulations can be affected to some degree by the pinning. Next slide, please. So this slide, again, I apologize, the graphs may not be terribly clear. I'll just run you through the very principal results. So the 5 graphs on the left, going from top to bottom, are the experimental data from the experiment. The corresponding 5 groups in the right-hand side column are results of simulations of this experiment run assuming that each local population is following a rigor map with parameter values roughly corresponding to the LH regime in which this population was done. In the graphs on the left, there are white circles which are high migration. There are black circles which are data from low migration treatments. And along the x-axis are the non-perturbed controls going from left to right, the block perturbation and the scattered perturbation. So first, if you just look at the 2 top graphs on the left, the topmost graph is the metapopulation fluctuation index. And what we do see is that the combination of high migration and the scattered perturbation, where the pinning can trickle down to all 6 subpopulations, that gives you the highest metapopulation fluctuation index, or in other words, the greatest constancy instability in that combination. If you look at the corresponding combination in the second graph on the left, which is a plot of metapopulation level extinctions, the entire set of 6 went extinct in the same generation. Again, extinctions are high for this one combination of high migration, 30% migration, and scattered pinning. So at the metapopulation level, constancy and persistence are behaving somewhat similarly in this experiment. But if we look at the third and the fourth graph on the left, the third graph is the subpopulation fluctuation index, and the fourth graph is the subpopulation extinction rate. Here, for example, if you look at the white dots under high migration from the middle going up to the right, there is an increase in high migration treatment in subpopulation fluctuation index when you go from block perturbation to scattered perturbation, your fluctuation index increases, constancy stability has come down. But if you look at what is happening to those corresponding 2 white points in the fourth graph, extinction rate is not changing. Similarly, if you look at the black dots on the third graph, from control to block perturbation, fluctuation index has come down. And again, if you look at the corresponding 2 black dots in the subpopulation extinction in the fourth graph, extinction rate has not changed. So at the subpopulation level, constancy and persistence are doing different things in response to the perturbations of the metapopulations. But at the metapopulation level, constancy and persistence are actually behaving in a fairly similar manner. So this again sort of emphasizes that there's one more layer of complexity depending on whether you're looking at the effects of perturbations on constancy versus persistence in local population level or global metapopulation level. The bottommost graph on the left is showing you the experimental data, the cross correlation coefficients. And what I will draw your attention to is simply the highest value, the rightmost white circle, which is the combination of high migration 30% and scattered pinning, where the effects of the pinning trickle down to all individuals, all subpopulations in the metapopulation. That gives you extremely high synchrony or correlation coefficient. And it is the combination of this high synchrony with the very high metapopulation Fi that you see in graph number 1 on the left, that combine as predicted by theory to give you a very high rate of metapopulation extinction for this combination of high migration and scattered perturbation, which results in high coherence. So indeed, the combination of high coherence or synchrony and high metapopulation fluctuation index can give you greatly elevated rates of metapopulation extinction. The simulation results on the right are just put there to show that you get fairly good agreement with the experimental results. The significance of this is that the simulations assume that the populations are following a simple model of population growth like the Ricker, which doesn't build in any species-specific biological detail into it. So this suggests that the results we are seeing on the left in the experiment are actually not due to some unique aspects of the biological system we use, but are more fundamental mechanisms, because they are captured even by a model that is a very simple population growth model. Next slide, please. Let me just say a couple of words on this, because we started by talking about Larry Muller's seminal 1988 Drosophila model. So some 31 years later, we have an improvement, which is the most effective model of Drosophila population dynamics yet. In that, this model actually fits data from not just 1 or 2 food regimes, but all 4 combinations of food regime, LL, LH, HL, and LL. It also fits data from populations that have been subjected to selection of varying kinds in the laboratory. Basically, it builds on Larry Muller's model. It builds in those 3 fundamental density dependent regulatory loops that we talked about, but it does this modeling in an agent based or individual based modeling framework. And this was work largely done by Sudipto Tung, who was actually an undergraduate intern through the Pope program in our lab, and then went on to do a PhD with Sutirth Day, who had interned and his PhD with me earlier. And so this is just to show you on the left, the top 4 graphs are the experimental data from 4 different food regimes. The bottom 4 graphs are simulated data from those same 4 different food regimes. Reasonably good capture. On the top graph on the right, each of these 4 clumps are showing you the distribution of population sizes along the y-axis in experiments on the left, and in the shaded bars is the corresponding simulation data from that food regime. So going from left to right, you have HH food regime experiment simulation, HL food regime experiment simulation, LH food regime experiment simulation, and LL food regime experiment simulation. The point I just want to draw your attention to here is not only are these simulations based on this model able to capture summary statistics like mean population size or median population size or fluctuation index or rate of extinction very well, they are also able to capture the distribution of population sizes across the time series really well. And this is something that previous models have not really been able to do. And subsequently, we have been using this model to do all kinds of really interesting things, looking at effects of different kinds of food regimes on all kinds of aspects of population dynamics, including some applications of population dynamics using things like sterile insect techniques and stuff like that. So this has opened up a lot of interesting new work. Next slide, please. So I'll just winding down here. Some of the other work we've been doing that I didn't talk about is the dynamics of heterogeneous 2-patch systems. So here we talked about metapopulations in which all local populations were either destabilized or stabilized. What happens if you have metapopulations or two patch metapopulations where one population is stable, the other is unstable, and so on? And we have done quite a lot of both theoretical and experimental work on that. We've also come up with so far the only real explanation out there in the literature as to why two coupled chaotic populations that are stabilized by low dispersal simultaneously also go out of phase. This was again done in collaboration with a summer student from IISER Pune, Vedat Goswami, who did some really cute work on this. And I just mentioned we've been looking at how life stage specific food levels interact with other ecological factors, mediating their effects on dynamics and stability. And a major line of work in our lab is also the evolution of competitive ability via different suites of traits. We briefly alluded to this, and this is actually a whole different story in its own right. And I put this picture down here just to show that scientific advances are cumulative. We began this story by noting that Larry Miller's work in the 1980s and 90s laid the foundations for our ability to then follow up doing the kinds of studies we did. Here is Larry Miller on the left during a visit to India in 2013. Next to his left is myself, and then Sutirth Day, who did some of the metapopulation work earlier. And on the extreme right is Shudipto Tung, Sutirth's student, who did the more elaborate Drosophila model that I just mentioned. Next slide, please. So some thank yous at the end. The middle picture here is of a very, very remarkable person, unfortunately no more, Professor Prabhakar Vaidya, who was one of the most intellectually sharp and interesting and nicest people I've ever met in my life. And much of whatever I learned about nonlinear dynamics, not having studied this formally as a student, was through a lot of discussions with Professor Vaidya. On the left top is coincidentally his daughter Gitanjali, who did a master's project in our lab. And she did some of the work on Drosophila populations adapted to crowding and their stability and dynamics. Top right is Neha Pandey, who is just finishing up her PhD. She's writing a thesis. She has done also work on melanogaster populations, the effects of adaptations to crowding on stability, and so on. Bottom right is Medha Rao, who is presently in her third year of her PhD. She is doing some work on the stability and dynamics of populations that are both subjected to life history evolution and adaptations to crowding, and trying to connect competitive ability, life history, and dynamics in a common framework. The center bottom is unfortunately again Dr. Snigdhadeep Day. He did his PhD with us and then was doing a postdoc in France, and very tragically died a few years ago at a very young age. So many of the studies I talked about, including the block and scattered perturbation, the LL metapopulation work, and so on, was Snigdhadeep's work. And on the left-hand side bottom are Rajanna and Muniraju, the sort of solid foundation of our lab. They help with all the experiments. They work with all the cleaning of glassware, the media preparation. I mean, without them, it wouldn't be possible to raise so many generations or so many populations for so many years. And my work over the years has been funded from DST, internal funding from JNC, my institute, and also a JC Bose National Fellowship from the Science Engineering Research Board of the Government of India. And thank you all very much for your patience, and my apologies once again for the mess up with the slide sharing. Thank you.
Sourdeep, you have to unmute yourself.
Oh, my bad. Sorry. I was just thanking Dr. Joshi for this excellent talk. And it was really interesting to know about not only the experimental details, but also the historical, some of the historical perspective on the studies of population ecology. Because sometimes we all get so much bogged down to like really narrow field of our experiments. It's really humbling to know the background and what a great body of work that you have. And I think it's for any budding scientist, it is fascinating to see someone so motivated about science like you, even after 2 decades of academic research. And again, thank you for this captivating talk.
Thank
I
you.
think we'll just take one question in the interest of time, and then we will directly go to the interaction. You can, for all the viewers, you can join us on the stream yet if you want to directly interact with Professor Joshi. We will just take one question from Sundaram Acharya. He's asking, he's wondering what is the influence of genetic variation in natural population in the evolution of population stability?
That's an interesting question. So I don't think genetic variation on the whole, in and of itself, has a direct impact on the evolution of population stability. But the genetic variation, in conjunction with the ecology of the organism, which is providing the selection pressures, obviously mediates how the population evolves in its environment. And the manner in which different aspects of the life history evolve can in turn have significant effects on population stability and dynamics. So the whole notion of how evolutionary change in response to the ecology that the organism inhabits in turn affects the population dynamics and stability is an interesting area, and a lot of work is just now beginning to get done on this.
All right. Thank you again. Thank you to all the viewers who have tuned in from different corners of the world. We hope you enjoyed the talk. We certainly did hosting it. Before signing off, I would just like to share the joyous news of us getting funded by the American Society for Cell Biology. And of course, this wouldn't have been possible without all the amazing speakers we have had so far, and all the amazing viewers and their great questions. And again, if you'd like to interact with Professor Joshi, please join our StreamYard link, and you can directly ask questions to Joshi. With that, I'd like to bid adieu to you. We'll see you on our next talk. Thank you.